An asymptomatic person releases virions in saliva microdroplets when exhaling air. These microdroplets can contain up to 500 virions. The microdroplets are randomly dispersed and their trajectory is directed by the wind. In open places, the microdroplets can reach up to 6 meters, but dissipate quickly. If you are 1.5 meters from an infected person, you can inhale certain microdroplets. If they enter through the nose, they find structures called nasal turbinates. These cause turbulence in the air we breathe, so when passing through the nose, the microdrops stick to surfaces. But before it can reach a cell, the virus has to pass through a protective layer of mucus. With the first proteins that the virion stumbles upon entering the mucus are the mucins. These filamentous proteins give the mucus its viscosity. The mucins form molecular networks that trap particles in the mucus, impeding their movement. But virions are very small and the spike proteins are highly glycosidated. These oligosaccharides repel each other due to their negative charges, and the virus moves slowly, slipping between the mucins. But then, the virion is attacked by proteins called defensins. These proteins contain a positively charged part and a hydrophobic part. When approaching the viral membrane, the defensins are attracted by the negative charge of the phospholipids. Then their hydrophobic part fits into the membrane and destabilizes it. This kills the virion, breaking it open, causing its contents to spill out. More virions keep passing the mucus barrier, and defensins proteins keep destroying them. But little by little these proteins are used up and more virions continue to cross the mucus layer. But defensins are not the only defense in mucus. It also contains complement proteins that are even more aggressive against pathogens. The amphicoelin protein binds to the oligosaccharides of the spike protein. Together, a protease called MASP1 is activated. It hydrolyzes and activates the protease MASP2. MASP2 cuts and activates the C4 protein in two parts, the C4a and the C4b. The C4b part is covalently attached to the virus membrane. Then C2 binds to C4b and is hydrolyzed by MASP2 in two parts, C2a and C2b, to form the C4b-C2a complex, or also called C3 convertase. The C3 convertase begins to massively hydrolyze the C3 proteins into two parts, C3a and C3b. The C3b parts get covalently attached to the membrane. C3b couples with C4b, C2a to form the C4b, C2a, C3b complex called C5 convertase. C5 convertase begins to massively hydrolyze the C5 proteins into two parts, C5a and C5b. C5b inserts itself into the membrane of the virion and binds with the protein C6, C7 and C8 to form a protein complex that begins to insert C9 proteins into the membrane and forms a pore that destroys the permeability of the membrane, and the virion is disordered until death. However, some virions outlive the mucus defenses and continue to pass through it slowly. The mucus also contains type A immunoglobulins that could end up eliminating the remaining virions, but the spike protein is almost completely covered by oligosaccharides that serve as shields that block the immunoglobulins. There are exposed places in the spike protein, but this virus is new in the world population and very few people contain immunoglobulins related to the exposed parts of the protein. Despite all these defenses, certain virions manage to survive the innate defenses of the mucosa and reach a less hostile second layer of mucus. Now the virions are about to interact with the cells of the nasal epithelium in search of receptors that help it to fuse its membrane with that of the cell. This was the immunity of the mucous membranes, a fundamental part of the innate defenses that detects and destroys most of the pathogens that try to invade our body. 3 virions managed to survive the mucus barrier. They randomly moved towards the cells of the nasal epithelium and landed on a goblet cell. These cells produce mucus and the mucin proteins of the last episode. Many different proteins are found in the goblet cell membrane. Most of them are used to detect outside signals, but none of them evolved to be the receptor of a virus. However, the virus did evolve to bind to different cell proteins that favorably position them on the membrane. With the first proteins that the spike protein interacts with are the heparin sulfate oligosaccharides of the syndecan proteins. The oligosaccharides of heparin sulfate bind to the positive domain of the spike protein. These proteins serve exactly as platforms in which various proteins bind to approach more efficiently the cell membrane. Advantageously, the coronavirus evolved to use it to its benefit as a co-receptor. Once it is anchored, it randomly approaches the cell membrane and subsequently binds with another protein called neuropilin 1. The function of this protein is to regulate cell growth and organization in tissues, but the coronavirus also evolved to bind with it and use it as a co-receptor, since it positions the virion at the perfect height of the membrane to bind with another protein called angiotensin converting enzyme 2 or ACE2. This protein is an enzyme that hydrolyzes the molecule angiotensin 1 to angiotensin 2. And it is essential in regulating blood flow and blood pressure. ACE2 is at the ideal height to position the spike protein on the cell membrane so it can anchor itself to it, so the coronavirus evolved to use it as the main viral receptor. But the spike protein has to be activated just at the right moment it is positioned on the membrane, because it only works once, and if the spike protein is activated before being positioned on the membrane, its energy gets wasted and the virion becomes useless. So how is it that the virus manages to activate the spike at the right moment? In an ingenious way, the spike protein evolved a cleavage site that needs to be hydrolyzed by a protease in order to be activated. A protein that participates in proteolytic cascades called transmembrane protease serine 2 or TMRPSS2 is the one that binds in the cleavage site and hydrolyzes it in two parts, S1 and S2. The S1 part remains attached to the ACE2, and the S2 part begins to fold itself, extending towards the cell membrane. At the end of S2 part there is a series of hydrophobic amino acids that anchor into the cell membrane. And this is why it is very important that it gets activated once positioned on a membrane. The spike protein keeps folding into a lower energy state and in the process fuses the cell and viral membranes. In this way, the inside of the virus ends inside the cell, and the genetic material together with the nucleocapsids floats in the cell cytoplasm. But this is not the only route that the coronavirus uses to enter the cell. It can also enter through endocytosis. Goblet cells produce huge vesicles filled with mucus and proteins. This constantly adds membrane to the cell. To avoid membrane overgrowth, the cell needs to endocyte the same amount of membrane that it exocytes. In this membrane recycling, several membrane proteins get endocyted, and since the virion is anchored to the membrane, it opportunely gets into the interior of the cell within a vesicle that is transported through microtubes by a protein called dynein. But this route is the more hostile of the two. The cell has designed a series of security measures to prevent the entry of pathogens. The cell has not been infected yet. The virus versus cell battle is just beginning. The mucosal barrier is formidable and stops a large number of pathogens, but the intracellular defenses are even more amazing. The vesicle that contains the virion that entered through endocytosis fuses with other vesicles until it becomes a huge vesicle that we call the early endosome. Vesicles from the Golgi apparatus arrive at the endosome and fuse with it, delivering different proteins such as proton pumps and up to 40 acid hydrolases such as proteases, nucleases, glycosidases, lipases, phospholipases, and sulfatases. Together these enzymes can destroy any pathogen. But these enzymes work best at a pH less than 5, and the pH in the early endosome is 7. To activate them at their maximum rate and begin to degrade the contents of the endosome, proton pumps gradually raise the concentration of protons inside the endosome, lowering the pH to 5. With a pH of 5, the endosome transforms into its mature form. The spike proteins separate from their receptors an acidic pH, and the acid hydrolases begin to degrade the macromolecules inside the endosome. But coronavirus virions contain two highly glycosidated membrane proteins, the spike and the M protein. The oligosaccharides protect the virion from lipases, proteases, sulfatases, phospholipases, and nucleases, but not from glycosidases. The glycosidases begin to hydrolyze the oligosaccharides of the virion, but this gives it enough time to try to escape the endosome. The spike protein is absent of oligosaccharides in its cleavage part, and the protease cathepsin L hydrolyzes the spike protein and separates it into its two parts, but the virion is not near the endosome membrane and is unable to fuse with it. The glycosidases finish degrading the oligosaccharide barrier, and the other hydrolases tear the virion to pieces, exposing the genome, which is also hydrolyzed into smaller pieces of RNA floating inside the endosome. The presence of RNA in the endosome only means one thing: the presence of pathogens. These pieces of RNA are detected by proteins that are only found in the endosome. Toll-like receptor 7 binds to single-strand RNA, and toll-like receptor 3 binds to double-strand RNA. When two toll-like receptors bind through their ligand, they form a dimer that can bind to other adapter proteins in its cytoplasmic domain. The dimerized toll-like receptor 7 bind to the MDA-5 protein, which acts as an adapter connecting proteins that receive signals from outside the cell to proteins that transmit signals within the cell. Then the IRAK4 protein binds to MDA-5 adapter protein, and IRAK1 binds to IRAK4, forming a protein complex called the Endosome. The IRAK4 protein is a kinase that is activated by autophosphorylation. Once activated, it phosphorylates IRAK1 and activates it. Then the TRAF6 protein binds to the Endosome, and IRAK4 activates it by phosphorylation. The dimerized toll-like receptors 3 bind to the adapter protein TRAF, and the protein TRAF3 binds to TRAF. TRAF proteins are ubiquitin ligases. They bind to UBC13 protein, which contains a covalent bound ubiquitin protein. UBC13 transfers its ubiquitin to TRAF. More and more UBC13 begin to transfer ubiquitin, creating a polymer that serves as a platform where different proteins anchor to be activated. The TAB1, TAB2, and TAK1 proteins form a protein complex that binds to the polyubiquitin, so they can be phosphorylated by the IRAK1 protein. The protein complex called NEMO, that is made up of IKK alpha, IKK beta, and IKK gamma, is also anchored to polyubiquitin to be phosphorylated by the TAK1 protein. Lastly, the TBK1 protein is also anchored to polyubiquitin and activated. Now everything is ready for what we call a signaling cascade, and appreciate graphically how a small signal from just two receptors can exponentially amplify the signal, activating hundreds of genetic regulators. From the protein complex of toll-like receptors 3, which predominate in epithelial cells of the respiratory tract, the TBK1 proteins begin to phosphorylate IRF3 regulatory proteins. From the protein complex of toll-like receptor 7, the IRAK1 proteins phosphorylate IRF7 regulatory proteins. The NEMO complex begins to phosphorylate inhibitor proteins of nuclear factor kappa beta. And TAK1 proteins phosphorylate MAP kinase proteins, and these phosphorylate P38 and JNK proteins. As we observed, two toll-like receptors activated dozens of signaling proteins, and each of them activates dozens of regulatory proteins. So for each activation level, the signal is amplified and hundreds or thousands of regulatory proteins are activated from just two receptors. Once the regulatory proteins are activated, they are imported into the nucleus to begin gene expression. But before the nuclear factor kappa beta enters, it must be detached from its inhibitory protein that prevents it from being transported to the nucleus. When the protein is phosphorylized, a ubiquitin ligase binds to it and transfers ubiquitins to form a polymer, but this time the ubiquitins are connected in a different way, forming a polymer with different shape to the polyubiquitin that we saw earlier. This polyubiquitin binds with a protein complex called proteasome, and its function is to break other proteins into small peptides. By degrading the inhibitory protein, the nuclear factor kappa beta is recognized by importins, which function is to import proteins to the nucleus, where they will begin to express antiviral genes to counteract the attacks of the virus. But at the same time that this happened, another virion introduced its genome into the cell by membrane fusion. As it is not connected to a motor protein, it is almost immobile in the cytoplasm of the cell. But entering the cytoplasm is not enough to infect it. The cell also has antiviral defenses in its cytoplasm that the virus must overcome in order to take control of the cell. The nucleocapsid proteins that are covering the viral genome begin to randomly detach themselves, leaving the genome naked. As it is not attached to the nucleocapsids, the genome begins to fold, forming loops that are essential to regulate its translation to proteins. We call this structure secondary structure of RNA. But to start synthesizing viral proteins, the genome has to randomly meet ribosomes. The ribosome is a complex of proteins and RNA so big that it stays almost immobile in the cytoplasm. So it is much more likely to meet with other smaller proteins like ribonucleases. Ribonucleases are enzymes that break down RNA that is damaged or must be discarded. Ribonucleases XRN1 degraded from the front in the 5' part, the dis3L2 degraded from the back in its 3' part. Cell messenger RNAs are not immediately degraded by ribonucleases. Messenger RNAs contain a molecule that we call cap in its 5' part, and this prevents the XRN1 from binding and degrading the RNA. In the 3' part, the messenger RNAs contain a series of adenines that also prevents dis3L2 from binding and degrading the RNA. Surprisingly, the coronavirus has evolved the ability to synthesize a false cap similar enough to evade the binding of ribonuclease XRN1 and not be degraded. The only difference is in a methyl group that is missing from the viral RNA cap. On its 3' side it also contains a series of adenines that prevent the binding of the ribonuclease dis3L2. The viral genome has managed to evade the first intracellular defense. But he still needs to evade more aggressive defenses. The RIG and MDA-5 proteins are very similar proteins that detect RNA of viral origin and are as important as toll-like receptors. The RIG protein detects false capped viral RNA, and MDA detects very long regions of double-stranded RNA. When either of the two is activated, the antiviral response and degradation of the viral genome are initiated. But as we have already seen, the viral cap is too similar to the true cap, and this similarity decreases the affinity with the RIG protein. Even so, some RIG proteins manage to bind to the false cap, and the RIG protein attach themselves to the viral RNA. When 4 RIG proteins bind to the RNA, they form a tetramer. Then the TRIM protein adds ubiquitins to stabilize the tetramer so it can bind to IPS-1 proteins. But the virion has a weapon to counteract the RIG proteins. The nucleocapsid protein that was introduced to the cell along with the genome is what we call the Swiss Army knife protein. Because it not only fulfills the function of packaging and protecting the genome of chemical damage, it also has several very important functions at different stages of the viral cycle. One of them is to interrupt the antiviral response, giving the genome more time to take control of the cell without being detected by the cell. This is achieved by binding to the TRIM25 protein, inhibiting its function and preventing it from adding ubiquitin to RIGs, blocking the activation of the antiviral metabolic pathway. But the cell is not so easy to tame. It contains another similar protein that is very effective against coronaviruses, MDA-5. This binds to regions of double-stranded RNA and forms a ring that surrounds the RNA. More and more MDA-5 proteins fold to the RNA to form cylinders. Every 4 MDA-5 join to form ring-shaped tetramers. These rings serve as primers that initiate the polymerization of filaments in the membrane of mitochondria. Transmembrane IPS-1 proteins found in the mitochondria bind to the ring of the MDA-5 protein tetramer, and more IPS-1 proteins begin to bind, forming a polymer of IPS-1 proteins that spreads on the surfaces of mitochondria. These filamentous complexes serve as a platform for the TRAF3 and TRAF6 proteins to bind. And as we saw in the last episode, the TRAF3 and TRAF6 proteins serve as platforms where other proteins join to be activated, and these in turn activate others, and thus begins the signaling cascade that culminates in the activation of regulatory proteins such as IRF3, IRF7, nuclear factor kappa beta, P38 and JNK. The nuclear factor kappa beta is removed from its inhibitory part through its degradation in the proteasome. All these regulators are imported into the nucleus to activate antiviral genes. The interferons. Interferons are messenger proteins that cells release after detecting the presence of viruses. They serve as alarms that alert neighboring cells to impending danger. In the event that a cell has already been infected by a virus, interferons also serve to rescue it and cure it of the infection, being of great help to prevent viruses from colonizing a tissue. Regulators that were activated by toll-like receptors and Rig-like receptors bind to importins, and these bind to the nuclear pore complex to be imported into the nucleus. Once in the nucleus, the JNK protein phosphorylates the regulator c-Jun, and the protein p38 phosphorylates the regulator ATF2. c-Jun and ATF2 bind to form homodimers. Regulators IRF3 and IRF7 form heterodimers, but can also combine with each other to form homodimers. And the nuclear factor kappa beta is made up of p65 and p50. Later, these regulators bind weakly to DNA and begin to travel through it, sliding down their spirals in search of the gene that codes for interferon proteins. The gene is inactivated on chromosome 9 by histones that guard and protect the information. At the beginning of the gene there are 4 regions to which the regulators bind strongly to activate it, and we call them cis regulatory sequences. Regulators P65 and P50 reach the second region and bind tightly. Regulators IRF3 and IRF7 bind to regions 1 and 3, and c-Jun and ATF2 bind to region 4. When the 4 regions are occupied by regulators, they form a complex called Enhanceosome. This complex binds with the protein GCN5, an acetyltransferase that adds acetyl groups to the tails of histones H3 and H4. Then a kinase protein also binds to this complex and adds phosphates to the modified H3 histone tails. Once the H3 histones are acetylated and phosphorylated, the GCN5 protein adds an additional acetyl group to them. Once the H3 histones have been modified, the BAF complex binds to them. This complex displaces histones to uncover a sequence called TATA box and activate the gene for interferons. Transcription factor 2D binds to histone H3, and this in turn is bound to TBP, which binds to the TATA sequence in DNA. At the same time, the P300 protein also binds the Enhanceosome complex. This protein helps attract RNA polymerase II and transcription factors, so they bind faster to genes and accelerate the reaction. RNA polymerase II begins to polymerize an RNA strand that complements the DNA information. Some proteins add a cap to the RNA on its prime 5 side. Then, different proteins bind to the tail of RNA polymerase II, and these bind to RNA sequences that divide introns from exons. Spliceosome proteins bind to these proteins and separates introns from exons. Finally, other proteins add a repetitive series of adenines, called poly-A tail. At the end, a mature messenger RNA is synthesized and is subsequently transported out of the nucleus to the cell cytoplasm. Here the messenger RNA meets the ribosomes. First, the 40S subunit binds to the mRNA and moves forward until it finds an AUG sequence and binds tightly. Then the 60S subunit binds to it and begins to translate the information from RNA to protein. The ribosome binds to proteins in the membrane of the endoplasmic reticulum and introduces the interferon proteins inside it. The new protein begins to fold as it enters the endoplasmic reticulum. Here oligosaccharides are added and chaperone proteins help the protein fold correctly. Once the protein folds completely, it takes its 3-dimensional form and binds to proteins in the membrane of the endoplasmic reticulum that form vesicles and transport interferon proteins to the Golgi apparatus. Here, the interferon oligosaccharides are modified and the protein maturation ends. Once mature, it is transported in vesicles by motor proteins to the cell membrane, where it fuses with it to be expelled to the outside. Interferons disperse in the extracellular fluid and reach neighboring cells where they bind to interferon receptors. After binding, the receptors phosphorylized each other by Janus kinase proteins. Once phosphorylized, they bind to STATs1 and STATs2 proteins, which are also activated by phosphorylation. Once activated, the IRF9 protein binds to STATs2 and the protein complex is imported into the nucleus. In the nucleus, STATs regulators activate a set of up to 300 genes, which drastically change the metabolism of the cell, making it an hostile hell environment for viruses. We call this metabolic state the antiviral cellular mode. In this mode, the defenses in the cell membrane are reinforced through the expression of transmembrane proteins called epitome. These prevent the membrane fusion of the virus with the cell, increasing the viscosity of the lipid bilayer. It also increases the expression of tetherine proteins, which anchor to the membranes of viruses, disabling their escape. In the antiviral mode, protein production is also stopped and cytoplasmic RNA is degraded, interrupting the viral cycle of any virus. All cytoplasmic messenger RNA is degraded by the expression of endonucleases that cut RNAs in two. The PKR protein phosphorylizes the ELF2 initiation factor, inhibiting its function, essential for the ribosome to find the AUG initiation sequence in messenger RNA. Proteins that attract cells of the immune system, such as dendritic cells, macrophages, and neutrophils, are also expressed. They also attract natural killer cells, which induce cell death on infected cells. These and many other metabolic modifications prevent the virus from infecting and replicating within the cell. But at the same time, it affects the normal function of the cell, affecting the normal physiological functions in that tissue. The other neighboring cells go into antiviral mode, increasing their defenses. This prevents other virions from infecting the tissue of the nose, and the organism ends up eliminating the remaining virions. This case represents a person who takes precautions by keeping his distance to a minimum of 2 meters. 5 minutes later, the bus waiting for the lady arrives at the stop. She gets on and sits in the middle. The bus has air conditioning and closed windows since it is a very hot day. This creates a current of air that circulates the bus continuously, and with each breath the microdroplets in the air increase and increase. The most affected people are those who are directly in front of the lady. But the bus windows are closed and the microdroplets slowly disperse equally throughout the bus. In this scenario, the amount of microdroplets is much greater. Each breath is like a microscopic rain inside the nose and throat. Thousands and thousands of viruses pass the mucus barrier and go towards the cells. In the next episode, we will begin to see the mechanisms used by the virus to overcome antiviral defenses and how it manages to infect and take over cellular metabolism.